Hi, everyone. It's my great pleasure today to introduce Ferdinando Pucci for a webinar hosted by us at Ariadne AI. So we've known Ferdinando for a while because through this great spatial biology work he's doing at OHSU. So Ferdinando earned his PhD from San Raffaele University in Italy and did postdocs at EPFL in Lausanne in Switzerland and also at MGH with Harvard University in Boston. And after spending a little bit of time in industry, Ferdinando actually went on to join OHSU in Portland, Oregon as faculty and has been an assistant professor for tumor immunology there since 2019. So Ferdinando and his team have co-authored 29 peer-reviewed publications in immuno-oncology and adjacent field. He's a co-inventor on 5 patents. I do want to point out that Ferdinando is currently looking for faculty positions elsewhere, potentially including Europe. So if you hear anything, feel free to reach out to him independently. And today, Ferdinando will speak about how he has reinvented an old method, good old IHC, for spatial biology and used it to link patient survival with lymph node B-cell responses and head and neck cancer. So Ferdinando, take it away. I believe I have to stop sharing though.
Thank you, Fabian, and thanks everyone for joining today. I will start sharing. The second screen. And then the presentation that should pop right there. Let me know if you see the right screen. Yeah, you should.
Yes.
Okay, yes, as Fabian mentioned, I'm junior faculty at Oregon Health and Science University in Portland. And today I will discuss how we somehow reinvented immunohistochemistry to make it more relevant for the current push into spatial biology. I have no conflict of interest to disclose. So this is an outline of what I will be discussing today. I will briefly mention why spatial analysis have picked up so much pace recently and how we reinvented immunohistochemistry, both in a sequential or multiplex way, but also the work we've done to help others not make our own mistakes and develop new panels for immunohistochemistry. Then we will have probably, if time allows, a small short break for questions on the first part, on panel development mainly. And then I will discuss some advanced analysis that, with the help of Ariadne, we were able to achieve to study B cells in the lymph nodes of cancer patients. So when you look at the spatial examination of tissues, this is really nothing new. It actually goes back more than 150 years to one of the fathers of pathology, Rudolf Virchow. With his book, Cellular Pathology, he was credited to be one of the first to put forward the idea that the tissue architecture actually is important. And this was pretty much at the same time when other techniques like staining techniques like H&E, hematoxylin, and eosin and the Golgi staining were invented. And so these were really to diagnose disease. What we are doing or trying to do today is actually to understand disease with the spatial examination of tissue at a whole different level, from imaging to spatial transcriptomics to AI-based interpretation of pathological sections. There is definitely a costly entry if you think about today instruments, technology, and costs. Here, just an example and a ballpark idea of how much having a spatial transcriptomic data set would cost from the instrument perspective, from the service, and actually even on a sample basis. And so it's always a trade-off, right? Do we wait for the cost to come down, or do we just get things done? And what I chose 5 years ago, 6 years ago, was to get things done. And so instead of waiting, we started looking into something that is very well established. It's a common procedure in basically any hospital in the world. It's based on formalin fixed paraffin embedded tissues. Detection is not sequence-based, but antibody-based. And you don't need any fancy microscope. You just need a bright field scanner to collect the information. And probably this is a technique that most of us are already familiar with. It is based on a primary antibody, which is recognized by a secondary antibody, which is conjugated to an enzyme. This enzyme can transform a substrate into a colored product that can be imaged by bright field microscopy. Traditionally, this has been this technique, immunohistochemistry, was known to have the potential to have up to 3 or 4 colors in multiplex somehow, meaning simultaneous detection. And this was based on the fact that if you had different enzymes conjugated to the secondary antibody, in this case, for example, alkaline phosphatase or horseradish peroxidase, you could add different substrates that would generate different color precipitate. Of course, the important part here is that the primary antibody is actually of different species. In this example, again, a rat antibody would be detected by the alkaline phosphatase secondary, and the mouse antibody would be detected by the horseradish peroxidase secondary. This doesn't come without limitations, of course. And there can be some level of color blending. For example, in this case, red and brown is not really, the best way to detect two signals, right? The enzymes may have different conditions, different optimal conditions for their activity. And so if you have to do it in one step, then you have to pick one. Generally, you would pick, something in the middle, and so both enzymes would decrease their activity, reducing the sensitivity, ultimately, the detection sensitivity. So instead of doing it all at the same time, there's been work before me, actually from the same department, that developed this technique sequential. And so sequentially, you could change the conditions one step at a time. And the sequential is based on this classical 3 steps of immunohistochemistry: staining and imaging, washing, and then detaching the primary antibody. And doing this over and over by, in a sort of a programmatic way to go back to step 1 and do the steps again. And this has been shown by Dr. Takahiro Tsujikawa, who was working in the same department as mine before I joined, in this paper in Cell Reports in 2017. And so you can see that now, with general average cost per sample, which is actually 100 times less what we have seen before for spatial transcriptomics, now things become much more feasible. Of course, also the throughput can be much lower. In this case, it was about in the tens. And we will see with the careful panel design, you can bring this up to about 30 or 40 possibly. So this is an important slide because it really tells you what is the structure of the sequential immunohistochemistry. So it is divided in cycles and rounds. You start from, of course, cycle 1, round 1, and you're here. The cycle is defined by the antibody stripping, which happens at the end of cycle 1. And the rounds are defined by the enzyme inactivation. And so you can see you have multiple rounds for each cycle. Each of these steps is one biomarker. And so in this case, for example, you have a panel that is made of 9 biomarkers. For example, you would start with, say, CD3, then you inactivate the enzyme, then you add a rabbit anti-CD20, and then again, image, inactivate the enzyme, and go on with a rat anti-CD68, and then at the end, you have the antibody stripping, which removes all these 3 antibodies at a time. Then you have a blank slate, and you come back with another primary, secondary, remove the HRP, inactivate the enzyme, primary, secondary, inactivation, and so on. So at this point, the big question is, how do we fill the markers in this order? Is that a random order is fine, or do we have to make some special considerations given the biomarker themselves, given the epitopes that we are choosing? What do we need to consider? And this is when we started thinking about, okay, the panel that was in the department before we joined was good for studying a certain tissue, the tumor microenvironment, and certain cell types, mainly myeloid cells and T cells. But we were interested in studying B cells and studying them in the lymph nodes. And so we wanted to develop a different panel. This is what we put together in this paper, in which we describe in details how to develop new panels for sequential immunohistochemistry. Among the things that we observed was that there is a different performance of the primary antibodies depending on which cycle they are used in. For example, CD66B, which is a marker for neutrophils in humans, if you use it on cycle 1, it's great, looks fantastic. If you use it at a later cycle, for example, cycle 9, you don't see anything. Same goes for CD141, which is a marker of dendritic cells subsets. You need to use it at an earlier cycle. Generally speaking, antibodies tend to work best at earlier cycles. You can find some targets that need more epitope retrieval, and so the first few cycles don't work that well, but they start working at later cycles. But that's more uncommon, I would say. And so technically, ideally, you would have to test each possible cycle for each marker. That's a lot of work, but we will see that that is actually not necessary with some considerations, and we will discuss all these considerations together later on. Something else that we noted is that you can have carryover of signal within each cycle. And this is generally happening when you have a very intense staining, because, for example, there is a marker like CD20, which is very abundant in your section. As I mentioned, these are patient lymph nodes. So CD20 is basically staining half of the cells in the section. And in this case, for example, we noted that at the next round, we went to stain for CD66B, the neutrophil, which are relatively rare in the lymph nodes. And we saw different outcomes for different patients. And so we thought, okay, this really looks like the same staining as CD20. And we thought, well, this is the same round, sorry, it's the same cycle, it's just the following round. That means that what has been happening in between didn't work in this patient. And what happens in between is the enzyme inactivation. And so we thought, well, if the enzyme inactivation didn't work, then you would still have the enzyme bound to the CD20, and so giving you the same signal in the following round. And to test this, we did 2 rounds of enzyme inactivation, a dual blocking of the HRP. And we were able to recover the real signal of the neutrophils by CD66B staining. I should say these are all the same sections from this patient, patient 1, and these are also the same sections from the second patient. And you can see that even with the 2 rounds of enzyme inactivation, you can still keep a decent signal for the sections that were actually able to work on the first try. Yes, this is because of, we think it's because of accumulation of enzyme due to the intense staining, and by inactivating it twice, it was enough. There is another issue that can happen that we observed during our tests. This was cross-reactivity. So this cross-reactivity is happening at the level of the secondary antibody. So let's go into this example here. Here we have one antigen, one marker, which was again a CD20 that was detected with a goat primary antibody. So to detect that, we use the rabbit anti-goat. And the red precipitate formed here. At this point, in round 2, we had CD3. CD3 was detected with a rat primary. And we went on to detect CD3 with a goat anti-rat. Now here you could think that there is some cross-reactivity because the secondaries may bind to each other, because the first secondary could bind to the goat secondary in the round 2. But this doesn't seem to be a problem. You can see that the signal, when made into a pseudo-color, you can see that there is really no colocalization of the 2 signals. There's pretty much no yellow signal here. What can happen is when the overlap of the secondary antibodies, the secondary reagent, is actually double, meaning that one secondary recognizes the other and vice versa. And so now at this point you have double binding. You have the secondary from the first round that binds to the secondary of another round, and then you end up having a lot of co-staining, of colocalization of the signal. For example, this happened when we did a round 3 with CD66, a marker for NK. Here we use the goat anti-rabbit secondary. And this reacted, was really complementary both ways with the rabbit and the goat. And so when you have these situations, you need to be cognizant to realize that if you have 2 secondaries that really match each other this way, then you have a much higher likelihood of cross-reaction. So in this part, we wanted to address the difference between antibody stripping and antigen retrieval. Generally, these 2 steps are considered the same, are done at the same time. At the end of the cycle, you have a stripping of all the primary antibodies, and that is also acting as an antigen retrieval step. which is why sometimes you have some markers that work best at later cycles because they need 1 or 2 rounds of antigen retrieval. However, some markers need to be retrieved, the epitopes of these markers need to be retrieved at a different pH. In this case, this was, it's not written here, but this is a BCL6, which is a marker of a germinal center B cells. We went to do the usual step, and we did the antigen, the antibody stripping, assuming that it was also working as antigen retrieval at pH 6, and that didn't work. We just had, in this panel B, we just had a fainter signal of the previous marker. So we did, first we did the antigen retrieval at pH 9. Then we did the antibody stripping to restore the pH of the slide. And then we were able to detect, oh, here it goes. And then we were able to detect BCL6 at a later cycle. In this case, you can notice that since we did an antigen retrieval step at this point, we had to do it twice. Sorry, since we did right, we did the antigen retrieval twice, so we had to switch up with the cycle twice as well. This is just a technicality. So overall, we proposed these considerations for trying to find a balance between doing all the possible combinations or save time and lower cost to find the best balance with the least amount of work. We do have an antibody database that is available to browse for everyone. Please check this paper, in which you will find a link to a website, Airtable, that has all the tests that we did, including those that didn't work. And so, in this database, we have more hundreds of conditions for tens and tens of markers. So we would start, if you want to develop your own panel, maybe you don't do immuno-oncology, but you do neuroscience or cardiovascular studies, you want to have your own panel to work on, you have a wish list. You can, we always recommend to identify potential alternatives in case you need to swap them. Then you can consult either our database or the literature to see if there is any information on which cycle these primary antibodies can be working, are likely to work. And so you can already start to fill these into the panel. It's important to identify conflicts early on, as we discussed, either cross-reactivity or carryover. Once you identify these conflicts, both for primary and secondary antibodies, you can already have some alternatives as a backup. You will have to do some tests for the unknowns, the markers that there's not enough information out there. And so it's always good to have some practice slides. And it's important to keep the practice slides as you use them, because you want to always have some practice slides at cycle 1, 3, 5, 10, so you can test the antibodies at a slide with a different cycle. It's important to understand also the difference between antigen retrieval and antibody stripping, because it's not always the case. It's important to know which of your biomarkers will need a retrieval at different pH than the classical citrate buffer at pH 6. And then once you have a good candidate panel to test, you want to be mindful of potential cross-reactivity. And so consider when putting a very abundant marker, in our case, a CD20, whether you want to put that one at the beginning of the cycle or at the end, for example. So in this study, we already had some quite interesting observations that we were able to make. And this was, we found that plasmablasts were interacting with a specific cell type. We were always found in close contact with a specific cell type. We were interested in plasmablasts because, of course, those are the cells that generate the antibody-producing. The final differentiated plasma cells, which are the antibody-producing cells. We were interested in B-cell effector functions, and so we were curious about which effector cells would be sensing the antibodies that the plasmablasts and plasma cells make first. And so we thought, let's see which of the potential effector cells, which would be cells that express FC receptors, are around the plasmablasts and the plasma cells. And so we scored the distance between each plasmablast and plasma cell, defined as a CD138 positive cell, and all of these other innate immune cells. And we observed that by far, plasmacytoid dendritic cells were the ones that were really in close contact with these plasma cells. And we thought this was an interesting observation that may have implications potentially. And definitely, given the interest on the interferon response and vaccine-elicited immune responses, this could be something that may be worth further investigations. So this is the general outline that we inherited from the sequential immunohistochemistry and the improvements that we believe we have provided in this study. I think we are doing good with time, so if there is any question, I would take a break here, and if any has any urgent question or I wasn't clear on any of the steps, I would encourage you to turn on the camera or unmute yourself and
Yes. Just to be clear, we do use the Q&A function of Zoom, so you will see the Q&A button, if you haven't seen it yet, at the bottom of your Zoom screen, and you can type your questions there, and we'll read them. That makes it easier for us to host the webinar as a video for on-demand viewing later. So I actually have a question. Maybe I could go first. So it's great what you showed, and I'm not sure a lot of people are aware that they can basically refer to your database to design panels, and how much work you've already done there for immuno-oncology. One thing that I've been wondering is, as you said, if someone were to want to use this on a different application, different therapeutic area, whatever, and they just wanted to make small changes to the panel, what are the odds that some small changes just going to work based on common best practices, or is it really something where you have to put a lot of work up front into getting any results out of it?
Yeah, thanks. That's a great question. Actually, if for example, you want to make small changes and study similar tissues, it would be much easier than starting from scratch, of course. Say that you want to focus on dendritic cell subsets, or you want to add more functional markers for your population of interest. Then in your database, in the database that we published, you can find a lot of information on how you can move the markers around. How far you can move out in later cycles, the markers that we already have, to make space to your own markers. I think that's a great point. That will make things much easier. So if I were to, develop a new panel based on that, say again that we want to focus more on the interaction between plasmacytoid and dendritic cells and plasma cell, plasmablasts. And then, I would remove some of the markers that are not needed, maybe T cell and B cell markers, maybe some other immune cell markers, neutrophils, macrophages, and then look at our database to find a way to move the markers that you keep at later cycles as far as you can, and then have space at the beginning to add additional markers for plasmablasts. For example, you could add different immunoglobulin, isotype and class, or plasma cells. You could add some or plasmacytoid and dendritic cells. You could add functional markers, for example, interferon pathways markers, and so on. Yeah, that's a great point. Thanks.
We did get a question in the Zoom Q&A. Do you independently validate the antibodies prior to the panel design?
Yeah, that's also a very good question. So generally, there are reliable websites that, commercial vendors that can provide, this is another advantage of this technique, right? It's so popular, it's so widespread that you can already find that type of information online available, even from the vendor website. And they can even tell you, which other papers have shown that have proven that they actually work, they are validated already. And so unless you have, non-commercial antibodies, for example, a colleague of yours has been giving you some aliquots of a clone that they made. So those need to be validated, of course. But for the vast majority of the antibodies that we used, they were already validated against to be very specific for the target in question. Yeah, good point.
Great, thanks. I think there are no further questions in the Q&A, so we'll have a few more minutes at the end anyway for more Q&A. Thanks.
Sounds good. Thank you. Okay, let's move on and look at the studies that we did in patient lymph nodes, specifically from head and neck cancer patients. So this is a subset actually of all the markers that we had. You can see the breadth of the panel that goes that covers both T cells, different lineages, and myeloid, and is of course focused on B cells and immunoglobulins. We used HPV status in the patients as a proxy for survival. As you may know, HPV positive head and neck cancer patients have a much better survival than HPV negative cancer patients. And we wanted to understand if there were different B cell responses using spatial analysis. So among the first steps that our friends at Ariadne took was to correct for distortions, and they generated these distortion maps. This basically, the sequential procedure that I highlighted has a lot of steps, right? Has a lot of washing, and so the tissue needs to be really well attached to the slide and not slide. But of course, some damage or micro shifts in the tissue can happen, and these can be corrected computationally in this step, this alignment step. This also aligns the different markers one on top of the other and allows to generate a stack of images in which each marker corresponds to one individual cell. You can see here an example of CD20 aligned to the initial hematoxylin staining. And so before the alignment and after the alignment, you can see how the nucleus and surface staining of CD20 is perfectly aligned after this step. Another part of the pipeline, of the bioinformatic pipeline, is to detect and mask areas that are either out of focus or that have some tissue damage or that are just unwanted, like red blood cells in blood vessels, or the non-tissue mask of the outside the tissue border, which is generally something that can have a lot of artifacts because it's non-tissue. And so this is all helpful to reduce the artifacts and the specific staining that usually happens in these areas, and focus only on the real staining. And this is done via machine learning. So here, the next step is the segmentation. This is a little movie that shows, I think, how well the segmentation, which was also done on a trained machine learning network using ground truth generated by one of the staff scientists at Ariadne. And you can see how really the methoxylin matches for each cell. And you know that the lymph node is a very packed organ. Is not sparsely cellularized. It's a highly cellularized network of cells with small round cells. And there are also elongated cells, as you can see here in this blood vessel. And still, the segmentation was able to capture both, which I think is a really good feat. Especially in the subclavicular region, you may have more elongated cells, and they can both be captured very well by this segmentation step. I think this is one of the hardest parts in spatial analysis. After this step, once the tissue is segmented, now, and all the markers are stacked in one on top of each other, then you can assign the intensity level for each marker to each of these segmented cells. And this ends up generating basically a file similar to a flow cytometry file. You can see here an example of the H&E section of the lymph node tissue, and you can see the digital flow cytometry file, the pseudo flow cytometry that allows you to gate basically the cells, for example here T cells or B cells, and show them back onto the slide like a virtual section. The key part to remember here is that each cell in the flow cytometry file is associated with an XY coordinate. So you know the location of each event, each cell that has the markers that you're getting for. And this is, of course, crucial for subsequent spatial, advanced spatial analysis. So one of the first spatial analysis that our friends at Ariadne did was clustering assessment. Here I'm gonna touch on a loose feet because, of course, I don't have a much background in these type of concepts, but I was able to understand by frequent interaction with the computationalists at Ariadne. And so I was interested in clustering because, of course, clustering is important for B cells. You know, in the B cell follicle, the germinal center is a cluster of activated B cells. So I wanted to understand if there was a difference in germinal center formation between HPV positive and HPV negative patients. The sensitivity score, what it does is basically compares the clustering of your cell type of interest, in this case, the green cells, with the background density of the cells, which are the gray cells here. And you know, you may still have, you know, for example, in this random distribution, if you don't look at the background gray cells, you can still think that, here there is a cluster of cells because they are more dense than the green cells here, right? But actually it's not. This is still a random distribution of cells. But if you have a non-random distribution and you compare to the background, now you can tell, yes, this is a much more dense cluster of cells as compared to, for example, here where there's no cells, no green cells. And so this is how we looked at germinal center B cells. These are actually dark zone germinal center B cells because they are proliferating BCL6 positive B cells. You can see here is the overview of the markers. CD20 highlights the follicle, the B cell follicle. And this is the T cell area right on top of them, of the follicles. Here CD20 is in blue and BCL6 is in red. And you can see that there's a BCL6 and Ki67 double positive cells, which are dark zone germinal center B cells. And by looking at the clustering of these B cells, we saw that there was a significant difference between HPV positive patients and HPV negative patients. Here in HPV positive patients, you can see that the dark zone highlight the classical split in the follicle, in the B cell follicle, in these lymph nodes. You have the light zone and the dark zone. That's how they define the two poles, the two polarity of the follicle. And this suggests that the B cell activity here is well organized. The follicle, the germinal center has a good organization, which can lead to a better immune response. And maybe that's why we thought that correlates with better survival in HPV positive patients. On the other hand, in HPV negative patients, who have almost half the survival, unfortunately, of their counterparts, dark zone germinal center B cells are very disorganized. You can see some look even bipolar on some sections. And this was quantified statistically. We could add, not just say, oh, it looks like this. There is a statistical significance because we were able to assess this clustering centrality score between the two groups of patients. Next, we did something that I think is very cool here. It's a recurrent neighborhood analysis, and it has its roots in language modeling, natural language modeling. So, as far as understood, in this natural language modeling, you have different entities, right? For example, in a book, you have words like ball, cat, blue. You have topics. It could be a book about sports, right? And you have the whole document with the whole book, which is probably a collection of topics. It could be some, I don't know, sports for kids, and then sports for adults, and sports for elderly. This modeling has been applied very recently, actually, in biology. And the equivalent is that cell types were treated as words. The collection, so for example, B cells or even germinal center B cells was a cell type that was treated as a word. CD40 cells was a cell type, and so on. The collection of cell types in an area was a topic. So for example, the B cell follicle was a collection of cell types. It contained both CD40 cells, some macrophages, some germinal center B cells, of course. And lastly, the neighborhood, which is what we wanted to study, was the whole tissue, the whole document, the whole book. And this is defined by which cell types are around a certain cell of interest. Even more in details, don't ask me to comment on this slide, but I just copy and paste from the discussion we had with the friends at Ariadne, is basically how each cell neighborhood is calculated. Here, for example, you have the cell of interest, this black cell here. You define a radius, and for each cell of interest, you will collect a number of neighboring cells which belong to a specific cell type, specific word. They belong to a specific topic, so they are a collection of cell types. And these, with all these different mathematical modeling, becomes basically a spreadsheet somehow that generates these recurrent cellular neighborhoods. By doing this, we generated 8 neighborhoods from 0 to 7 in our sections of patient lymph nodes. You can already see that the bright green neighborhood is the B cell follicle. And just next to the B cell follicle, you have the T cell area, which is this purple one. And I think this is just amazing. In between the two area, which is the classical, the B and T areas are something that medical students learn, in their pathology and anatomy classes. But here in between, this algorithm, this analysis, spatial analysis, was able to identify 2 layers of that represent different neighborhoods because they are composed of different topics and cell types. And by doing this analysis, we saw that in HPV positive patients, there was an increase in BT cell interactions within the follicle. And you can see this was extremely significant when looked statistically. And again, this is something that you can add a p-value, not just a qualitative readout. HPV positive patients had a much closer distance between B and T cells in the B cell follicle, suggesting that this interaction, these 2 cell types maybe are interacting more often, again, to generate a better immune response. At the border zone, the one that was highlighting before, the blue and the orange. Oh, actually, this is the orange, the recurrent cellular neighborhood number 5, the orange. So the immediate border zone to the follicle, we saw also a very significant increase in interactions between CD4 and CD8 T cells. Again, I think this is just amazing. Some of the HPV negative patients didn't even have any of these interactions. And this, I think, tells a lot of how the immune response from the B cell can transfer to the CD8 T cells. Again, this is mostly correlations, right? Because we are not making any change in the patient, or we're just studying the sample as they are. But they suggest that with this type of analysis, advanced spatial analysis, we can have a much greater insight in the biology that is underlying to, as I said at the beginning, to really understand the mechanism of disease. These mechanisms, though, need to have some in vivo modeling in mouse models to be really confirmed. And this is what we did here. In this mouse model, we studied the interaction. We wanted to confirm that the B and T interactions were actually important. They had an effect on tumor growth. And so we used a mouse model that is a head and neck model of cancer, is a squamous cell carcinoma. It's called MOC2. And it's known to be resistant to checkpoint therapy. They don't respond to checkpoint therapy. And it's also known to be extremely immune suppressive. You can see on the control group on the left that there are very few T cells in the inner area of the tumor. And the T cells are just stuck on the outside, just like in patients. When we did interventions, I won't enter into the details, but we did interventions to trigger B and T cell interactions. At this point, we achieved strong activation of the B cells in the lymph node, in the draining lymph nodes, and we achieved a tumor reduction and a massive increase in infiltration of CD8 cytotoxic T cells inside the inner mass of the tumor. Interestingly enough, CD4 T cell distribution at the tumor site was still unchanged. They were still mostly at the edge and somewhat found in the middle, but the biggest difference was definitely on the CD8, and this was quantified here, CD8 TIL. By weight, again, the tumors were significantly decreased, and there was a lot of changes in the draining lymph nodes. For example, we were able to observe increased proliferation of tumor-specific CD8 T cells and expansion of the so-called stem-like CD8 T cells, which are lately been associated in patients as well to better responses to immunotherapy. And even in circulation, we saw effects. This effect pointed to the fact that we actually were triggering epitope spreading in these models by observing specific antibodies in the circulation and by observing other B cells in the lymph nodes that were being activated. And so in summary, I hope I have shown you that sequential immunohistochemistry can be a cost-effective strategy to design panels for spatial analysis. We went through some of the pipeline that goes into performing spatial analysis, and we discussed advanced strategies like clustering and neighborhood analysis as applied to the study of B cells in the lymph nodes of cancer patients. I would like to mention the most important slide, of course, my lab, including previous trainees and alumni. I would like to thank my primary appointment in the otolaryngology department and people who developed the sequential immunohistochemistry, including Dr. Sujikawa and Lisa Cousins. I would like to thank my collaborators, both in other institutions, at government labs, and especially at Ariadne. In particular, this study has been possible with the help of Mitien, but one for the recurrent neighborhood and cluster analysis. Mitien really helped with machine learning and the initial setup of the pipeline for the lymph nodes. Tozing did also a lot of work for the lymph node and tumor microenvironment to train and to provide the ground truth. And Fabian, Lucas, and Adrian for trusting me and all the samples that I was sending them with weird requests. I would like to thank my current funding from American Cancer Society, which just started. And thanks again for hosting me and for attending this webinar.
Yeah, thanks, Ferdinando. That was great. Again, we do still have a few minutes for questions and answers. If you have any questions, feel free to use the Q&A feature in Zoom, which allows us to host the recording of the webinar for on-demand viewing later. If you write the questions there, we'll get a pop-up notice and we'll read them for you. So actually, I still had another question. Like after seeing how you really bound it all together, basically from the methods part to actually doing real biology with it in the end. So coming back to one of the first slides you had about trade-offs that you're making, do you feel like how maybe could you explain maybe how you would make this trade-off for the next project? Would you still go with this method? I mean, you've talked about all the labor intensity of the method, but also the flexibility of using off-the-shelf antibodies that are validated and well understood. So those are certainly things that sort of point in different directions. But like, if you had to do it again, would you still do it like this?
Yeah, it's a great question. I think, well, I would, because you know, we have all the expertise and the setup, you know, but I would also consider other options as well, that maybe, you know, as time goes by, you always have to reassess your premise and assumptions. I think the fact that this technique is affordable and accessible is still very valid. If you really need a high throughput, of course, that's already a no-go, right? Because you know, here you can probably get by up to maybe 50, 60. So it's still a decent throughput for most applications. And it's something that, you know, it's more manageable from the analysis side as well, because, you know, when, for example, we do single cell sequencing, like many labs, and, you know, the massive amount of data, and it's also cryptic into the objects of the data sets. You know, you cannot just look at it like on a plot, on a flow cytometry plot, like, you know, maybe I'm old school. I like to watch the image of a slide, you know, 4 colors maximum, or a flow cytometry plot. But, you know, the accessibility of having tens of markers instead of thousands of markers makes it also more accessible. And so if you think you would be okay with a few tens of markers, you know, in the 30 to 50 range, I think this is still a good choice. Plus, nowadays there are machines that can automate a lot of these steps.
Right. Yeah. Actually, that was going to be a follow-up question that I had. Like, how much of this could you actually automate, or can you now automate?
Yeah, I'm not just talking about the old, you know, Ventana machines, which can increase a bit the cost, but still automates it. And by the way, a lot of these Ventana machines are already found in many pathology departments, so you wouldn't have the upfront cost of the machine, just the cost of the consumables. But now there are other machines as well, and you know, I won't make names, but I know there is another company in Switzerland that makes those type of machines, both for immunohistochemistry and also for immunofluorescence approaches, multiplex sequential immunofluorescence approaches. And so with those machines, which you know, are getting more and more popular, more and more institutes are acquiring them. You would already have access to a lower labor cost, and that would largely compensate for the slightly higher per sample cost.
Yeah.
Yeah.
Yeah, excellent. So I think we're just about out of time. So I believe that if you're registered for, I mean, obviously you're here, so you've been registered for the webinar, you should be getting an email with the on-demand viewing link. Feel free to spread that widely to your colleagues and friends and so on. It will be available basically for the foreseeable future. So you can really watch it at your own leisure as well. So with this, I'd like to really thank you once again, Ferdinando. That was fantastic. And
thank you for having me.
Yeah, and I hope we'll be able to do something like this again in the future.
I hope so too. And thanks everyone for joining.
Yeah. Thank you. Yes.
Thank you, everyone. Have a good day.
Bye-bye.